thank you very much for the invitation to be here and the very kind words, if slightly distorted from the truth, but I still appreciate the kind words. It is a really great pleasure to be here. It's amazing to be here in the shadow of some of the mountains I like to roam around in my spare time, so it's a great place to be. As Rachel said, today I want to talk about, really mainly about cancer and the origin of cancer in terms of what it is, where it comes from and why it is so common in our population. I do that, and I'll spend some time on that because I think it's very important for us to kind of understand that, because that knowledge brings with it a certain degree of calm, and we understand the origins. We start to think of it less as being a foreign invader trying to attack us, but as more as a natural consequence of our complexity as being human beings. And then I'll talk a little bit about some of our research that we're doing at the moment on new diagnostic tests for cancer. And then I'll talk about the general advances in the field of cancer research and how that's impacting on treatment for patients with the disease. So some of you, especially some of you men around my age, will know who this fellow here is. So complex organisms, so humans, anything with more than 2 cells to rub together, tries very hard to avoid cancer. And the way that's done is by several mechanisms. One is by immune surveillance. So we have an immune system which is very good at recognizing abnormal cells and trying to destroy those cells. There's an incredible capacity to repair damage to our DNA or our genes inside every cell of our body. The cells have very complex machinery to detect damage and stress, and therefore control the risk of cancer developing by stopping cells from dividing if they think those cells aren't functioning well. We've got numerous enzymes which detoxify carcinogens from the environment, so to kind of disarm those nasty chemicals. And most cells in the body have a limited time span for life. They only live for a few days, 3 to 5 to 10 days. And which means if they accumulate DNA damage or mutations, that they're going to die anyway, so those cells will not become cancerous. So it's been, through evolution, there's been an extraordinary amount of effort has gone into making us quite robust to the risks posed by cancer. So what could possibly go wrong? We have all these amazing systems to stop us getting cancer, but obviously we still see a lot of it. It's remarkable that snakes get cancer. Fish get cancer. So up behind me there, the top left is a melanoma in the snake. There's a melanoma in a zebrafish in the shot beside it. Obviously a brain cancer in a fly in the shot beside it. Then also more remarkably, shellfish get cancer. I couldn't show you the insides of a shellfish, but that shellfish get cancer. Coral get cancer. So that shot there's a coral in the middle. That piece of the coral to the left-hand side, that is coral cancer. Worms get cancer. There's a round worm there with a lump on its shoulder. That is a tumor in a very simple type of worm called C. elegans. Sharks get cancer. I know you've been told by the people who sell you shark cartilage that they don't get cancer. They do get cancer. There's a shark with a cancer on its jaw. And even dinosaurs got cancer. So the shot on the far right is a fossil of a bone tumor from a brontosaurus. So cancer is not only present in every single organism which has more than 2 cells to rub together, it's also a very old disease, so it goes right back into the histories of time. In humans, there's 15 million new cases per year, and again, that goes a long way back. So we have prostate cancer has been confirmed in an Egyptian pharaoh dating from 250 BC. The earliest known confirmed case of cancer is again a prostate cancer from a Scythian king, which I believe is somewhere in Siberia, from 700 BC. So again, the history of cancer goes right back, both in an evolutionary sense to simple organisms, but also in the human history right back in time as well. So why is it so common? We've got all these amazing tricks, such as the immune system and DNA repair damage mechanisms inside cells to repair the risk of cancer. So why is cancer still so common? And it comes down to one number, 37 trillion, which is the number of cells in the average human body. So it's simply a numbers game. Every one of us has about 37 trillion cells. And those cells have the capacity to develop cancer. So within every single cell, there is the entire human genome, which encodes about 20,000 different genes. And those genes do everything there is about a human's capability, how a cell divides or grows or invades or moves. Every feature of a human's behavior and biology is encoded by these 20,000 genes. And so there's 37 trillion cells out there with all this incredible capacity to do things. So the manual for cancer is embedded in every single cell, and we have 37 trillion cells, and therefore I think there's a recipe for things to go wrong. So cells which are starting to become cancerous acquire various tricks, and they do that because they're really playing with a full deck of cards. They can use every trick that is available to a human being. So for example, a cancer cell can acquire the ability to migrate or move just like cells in a developing embryo do. They can move in and out of blood vessels just like immune cells do. They can coax blood vessels to grow in towards them to provide a supply of oxygen, just as we see in repairing tissue. They can reduce their reliance on oxygen, just like cells which are starved of energy do as well. They can lengthen their telomeres, so these little pieces on the end of chromosomes, to make them immortal, just like stem cells do. They can divide very, very rapidly, just like cells in a healing wound do. They can pump out drugs, like kidney cells do. And they can hide from the immune system, like cells in the colon do. So all these tricks which a cell acquires as it becomes a cancer are just normal features of a normal body, but because the tumour has access to the entire genome where these capabilities are encoded, it can do whatever it wants, providing there's the appropriate evolutionary force behind it, and I'll explain that in a moment. So because cancer is not different from a normal body, it's simply an acquisition of normal capabilities put into one place, into one group of cells. It means there is always collateral damage when you try and treat cancer. It is not a foreign invader. It's not like a fungus or a virus or a bacteria which is foreign, and therefore you can treat it with a very powerful drug which only kills that organism. When you treat cancer, then you are affecting processes which are normal to a healthy person as well. And so there's always going to be collateral damage. So how does the cancer choose the processes it needs to develop and grow and invade and become a threat to our lives? It's really just two steps. The first step is that DNA damage. So random DNA damage in our genes can alter the activity of genes. Okay, so that's just a random effect. There's various causes for that, which I'll explain in a moment, but random DNA damage can alter the activity of genes. So that's the starting point. And then the critical point is natural selection, just evolution. So the same process of evolution which Darwin described when talking of the finches on Easter Island. When we talk about the development of the human species, we talk about evolution and the power of natural selection. It's identical in development of cancer. Mutations occur in cells by chance, and then if those mutations by chance have occurred in a gene which is involved in cell proliferation, cell migration, cell movement, then there will be selection for that effect, and that cell will survive. It'll do better than its neighbouring cells. It will begin to grow and build and migrate. Simply a process of natural selection, just as we see through normal evolution. So what causes the DNA damage that is the starting point for all this? Well, there are really four things which cause DNA damage. One are environmental carcinogens. So nuns smoking cigarettes are at great risk for developing cancer. So carcinogens is one. Another example of a carcinogen in the environment is the photograph below that, which is a chimney sweep from London from many, many years ago. And not surprisingly, these guys developed cancer of the scrotum at a very, very high rate because they had extremely high exposure from carcinogens from the coal fires to their nether regions. And so they developed a very high rate of scrotal cancer. So it's the first cause of DNA damage. So these carcinogens, they cause damage to the DNA. It's random damage. but then it provides the basis for natural selection later on. Some bugs also cause DNA damage. So we know about the viruses which cause cervical cancer, papillomavirus. There's also a bacteria called Helicobacter pylori, which infects the stomach and starts the process of forming cancer of the stomach in some individuals. The third reason is inheritance of faulty genes. And so a few examples there. You'll all recognize Angela Jolie from a fairly well-known breast cancer family. Beside her is Napoleon Bonaparte, who came from a stomach cancer family, and that's apparently why he held this pose, not because he looked dashing, but because he actually had quite a sore puku for many of his latter years. And a shot below that is a woman called Pauline Harawira, who's from the first family we worked on in New Zealand with inherited stomach cancer. And so this is a family where over a period of about 30 years, they had 25 deaths from stomach cancer from people as young as an age of 14 years, an average of about 30 years of age. And so incredible stories of cancer in family caused by genes being passed down through the generations. The fourth reason, and probably in all, the biggest reason to cause DNA damage to lead to cancer, is bad luck. 37 trillion cells in the average human body. Each cell contains 3.2 billion letters of DNA code. Okay, so that's your entire genome is 3.2 letters of code, which encodes 20,000 genes which do all these functions. And then just by chance, there are 1 to 3 random errors occurring every time a cell divides. So when a cell divides, it has to copy 3.2 billion letters of DNA code, and mistakes occur by chance. Despite all the efforts of the cell to repair damage, which does very, very well, an error rate of 1 to 3 errors per 3.2 billion letters of code is pretty good, but it's still enough. When you consider there are 37 trillion cells, you've still now got a mutation rate which is high enough to start to become a threat. And so in time, with this background mutation rate in so many cells, you will get mutations in genes which are involved in cell proliferation, cell migration, cell survival, and those mutations will enable those cells to divide more rapidly, to move, to migrate, and effectively to become a cancer. So to put that all together in this slide, we have various environmental exposures such as diet, viruses, radiation, carcinogens. We have bad luck, we have inherited genes, all lead to genetic damage. Most of that damage is just repaired or of no great consequence. Just a cell may suffer and may die, but a proportion of those mutations occur in genes involved in cell proliferation, cell migration, cell survival, and those mutations will then undergo natural selection so that they will grow in number in the population, and you'll find the development of a tumour has occurred. So it is a process of chance and a process of natural selection. So really, cancer is inevitable. The wheels will fall off at some stage in many of us. But it's important that despite it being inevitable, we have to do something about it. We cannot accept it. And we cannot accept it because many people in our world do not get a fair crack at life and all its various stages. And maybe you don't get to spend much time with your grandchildren, or you may not be there for your daughter's wedding. You may not get to push a swan into the back of a car to live life to its most rich. But regardless, everyone deserves a fair crack at life. And the problem with cancer, even though the rates will always be there, we'll always have cancer in our lives, we have to do what we can to resist and to fight it, because we want everyone to have a fair crack at life and all that it brings, even if it's only as bad as putting a swan in the back of your car. This is a photograph I took of Chris Jackson a year or two ago. Natural selection is very, very powerful, but it is blind. It's a blind process. And so we shouldn't give up. We shouldn't say, oh, well, this is too powerful for us, too clever for us. It's not too clever for us. We can't out-muscle it, perhaps. We can certainly, I think, outthink it. In the next few slides, I'll try and explain how we're going about our research to try and outthink this power of natural selection, which has given us so much cancer in our population. So I'll give you some causes for optimism. The first one I'll talk about briefly is the track record of research so far and the work of clinicians such as Chris so far. I'll talk about some new tools for risk prediction, which are really useful, helping choose the right treatment for patients. New tests to better assess patient response to drugs, so we can make sure patients are getting the right treatment for their disease. We have a much better understanding of the molecular basis of cancer now, and that's really driving our design of new drugs, which are going to be extremely useful in the coming years. We're starting to develop really exciting rational drug combinations, which are going to be, again, very useful. And also of great note is the arrival, or perhaps the maturation to some level, of the technologies called immunotherapy. So I'll cover off each of these ones one by one, each of these examples one by one. And just, keep in mind the photograph up there, that's how cancer treatment was not so long ago, and where they're measuring how far away the patient should be from the radiation beam using a ruler, and the man taking notes in the lab coat and the woman offering support. That was how it was not so long ago. So we have seen vast improvements, obviously, since those times. Right, a couple of graphs. I've only got, I think, 2 graphs, so please bear with me. As a scientist, I love graphs, and so I cannot leave them out of my talks, so I'll show you 2. And they're complex, they go different directions. But the first one is the 5-year survival for children aged between 0 and 14. And so it's on the axis up, it's the percent survival. And so it goes from bottom left, 1966 to 1970, and only out as far as 2005. So there's about 10 years of data missing from this slide. But look at the improvement in survival in the last 30-odd years. So each of those cancers has been a vast improvement in survival. And you look in particular, I think it's the blue line there, which is for leukemia in children. It went from having a less than 10% survival rate to now, in this slide here, up to about 80-odd%, and I believe today it's over 90%. So there has been remarkable progress for certain cancers, particularly kidney cancers, in their survival for long term now. Now, in adult cancer, I've turned it around here, so don't get concerned it's going the other way. So I've turned it around. This is now mortality rather than survival. So just to be confusing, I've turned the whole thing around. So this is mortality from colon cancer. And the 3 graphs are males, females, and everyone together. So don't worry about the colours, but notice the trend. So it goes from 1971 on the left out to 2010 on the right. Notice that trend, that drop in mortality in patients with colon cancer. And it's the same with every other cancer practically. With breast cancer, a very, very similar trend downwards. So a steady decrease in the mortality from cancer, and that is just through numerous incremental improvements in how patients are treated. Slight improvements in the drugs, improvements in how the drugs are delivered to patients. And it just goes on and on and on. And so things are trending in the right direction. Now this is probably my most complex slide, but again, I can't resist putting up the odd complex slide because this is actually really cool technology, and I will go through it in detail, and we'll stay here till we all understand it. So ring up the babysitters, tell them to stay on for another hour or 2 because we've got some work to do. So this is a test which is called MammaPrint, and it's available. I don't think it's funded in New Zealand yet, but you can get it if you've got the money to pay for it. And it's trying to predict what will happen to women who have been diagnosed with breast cancer, just localized, so no lymph node involvement, so just localized breast cancer. The question is, will my cancer come back, doctor? I've had it removed, I've had some treatment for locally, will my cancer come back? So I'm sure Chris might have some thoughts on this, but a question they will get all the time. So what's going to happen next? And so it's a hard decision for the oncologist to kind of comment on that. So this test has tried to address that question. In fact, it has addressed that question and has achieved some really startling results. So what we have here is the rows of little squares is data from individual patients. And the ones above the line in the middle are patients whose cancer did not come back within 5 years, did not spread in 5 years. And the ones below the line are patients where the cancer did spread in 5 years' time. Okay, and so that's your 2 main groups looking sideways across that figure. The columns are genes and the activity of genes, whether the gene is turned off or turned off in their primary cancer. So their primary cancers have been removed and analysed in the lab for the activity of 70 genes and worked out whether they have high or low expression. So if it's low expression, so low activity, it's a green box. If the gene is active and turned on, it's a red box. If it's black, it means it's kind of like somewhere in the middle. So what you see there is an intersection between those two colours. So look at the top panel, the patients without spread, without metastasis. Most of those genes are turned off, they're green, they're not turned on in those patients' cancers. The tumours, the genes to the left-hand side, to your left-hand side, a lot of those genes are turned on, so those genes are active in the patients where there was no spread. But you look at the patients below the line, the whole thing is roughly flipped over. So it's a wee bit kind of patchy, but the basic principle was there. The whole thing has been flipped over. The patients who had spread in 5 years had the genes on the left turned on, and the genes on the right were more likely to be turned off. And so this gives a great capacity to kind of look into the future and make some prediction about whether a patient's cancer is going to return or not. And therefore, the patient can receive, if they fall into the group below the yellow line, they can receive more aggressive treatment because there's a very high chance that their cancer will come back, so they need to have more aggressive treatment early on before there's obvious disease development or progression. So that's the type of tool that we're starting to develop in labs around the world now and increasingly coming into clinical practice. One which is very, very important to us, which is called circulating human DNA. And this, again, is a tool to better observe whether drugs are working on individual patients. And it's a really simple idea. The idea is that as a cancer grows, it will sometimes, its own DNA will fall out of the cell, or the cell will die and the DNA will be spilt into the bloodstream. And so a cancer patient will have DNA in their bloodstream which is derived from their cancer. And you can recognize it's come from the cancer because it contains mutations in the DNA sequence of genes known to cause cancer. So by taking a blood sample from patients with cancer and looking at the gene sequences in their blood sample, you can determine whether a patient has cancer to a certain extent, but certainly you can work out whether the cancer is responding to treatment or not. By measuring the amount of circulating human DNA in the bloodstream, you can determine whether the patient is responding to treatment or whether their cancer is growing or shrinking. So this is a technology which we think is going to really be very, very important. I'll give you an example of how it has been used. This is done by a group in the UK a few years ago now, and it's an example which I think very nicely illustrates the power of this technology. So here's a patient, a woman with breast cancer, and she received 2 drugs. So you see down the bottom there, there's the time and days out to about 2 years. And she received a drug called epirubicin for about 1 year, and then there's a gap of about 100 days where she received no treatment, and then she started on another drug called Taxol after that. And below that, in blue there, there's a CT scan result. So you can see the CT scans saying, well, it's been progressive disease, it's now stable, hasn't grown very much, and eventually it thinks that the disease has progressed, then it thinks it's stable again. And so the CT scans, the standard technology to kind of look at disease progression, is saying, yeah, there's a bit of a change, it's got a little bit better, but it's quite a blunt tool and hasn't really captured the tumour's behaviour. But if you analyse the circulating tumour DNA, you see quite a striking pattern which differs from what you can see with a CT scan. So the circulating tumour DNA is shown by that black line there. So you see the patient has started treatment with epirubicin, levels have fallen away really, really fast to practically undetectable, down to about 0. And then after about 6 months, the tumour started to climb back again. But the CT scan couldn't see that. It still thought, well, it's still doing okay. And the treatment was continued for another 6 months in that patient. And even though the patient was getting no clinical benefit from that drug. So it was a futile treatment. So you can see, and it's a similar story with the second drug, the Taxol, afterwards. So you can see here the potential benefit for this type of test. You can very rapidly, with a simple blood test, determine whether a treatment is working in a short period of time. You could then, in principle, start to try new drugs and give them a shot for a while and see how they go. Chris might want to comment on this, because I have these ideas in the safety of my office or my lab. Chris has to kind of deal front line and actually make decisions in front of the patient, so he might want to comment. But to me, there are a number of options around how you might better treat patients. So for example, you could look at how you could reduce the dose the patient is receiving to reduce the side effects, because you can see whether the tumour is getting bigger or smaller. You can keep a close eye on that with a simple blood test. And so I think clinicians are often anxious about reducing drug doses because the cancer might come back and they might it might escape or get too big to be controlled. But if you can look at a blood test and get data every few days if you want to, you can very rapidly see whether treatments are working or not, and whether changes in drug levels are effective or have caused things to get a little bit worse. You can certainly stop futile treatment, like that woman there with epirubicin. You can stop that early on and transfer the patient to something which might be more useful. You can think about better ways to have maintenance therapies, where you keep a patient on a low drug dose just to kind of keep an eye and keep the tumour at bay for as long as you possibly can. Previously, very, very hard to do and very risky to do, I think, because you did not have a tool which was sensitive enough to observe changes in tumour size at short notice. And I think in time it'll be probably used as well as a clinical trial endpoint. So rather than looking at overall patient survival and trials of new drugs, it might be that you can just use circulating tumour DNA as a way to get a very quick readout on whether a drug is working or not. So it might speed up the time required to run clinical trials and reduce their cost as well, which obviously will be very useful. So Healthier Lives, the big banner on the left-hand side there, is funding a project for Chris and myself and a professor in Auckland called Chris Print on circulating tumour DNA in New Zealand. So what we're trying to do is currently we've got studies going for colon cancer, for breast cancer, and starting soon for stomach cancer and also paediatric cancers. And we hope in time to kind of establish this technology in New Zealand so it becomes routine practice for all cancers in New Zealand. So Healthy Lives has funded this project, and Healthy Lives is one of the national science challenges. So it's a new form of funding the government has provided for research, where the goal is to actually not just do some research for a while and get some interesting results and publish them. It's actually to take the research through to the finish, through to the end point, and get the research implemented into clinical practice. So we're very grateful to the Healthy Lives National Science Challenge for giving us the funding to make circulating tumour DNA a reality in normal clinical practice in this country. And they are, at the moment, I have to tell you this, at the moment they are looking to secure funding for the next 5 years from the government, and they're looking for feedback. So if you want to have a look at what Healthier Lives does, what research it supports, feel free to go to their website and also make some comments on whether you think the research direction of the Healthier Lives is correct. Again, as Rachel mentioned earlier on, increasingly we realise as scientists, as researchers, we have to be better linked into community and need more feedback from the community. So we'd appreciate if you had a look at that website and gave us your thoughts on whether the research proposed in cancer, for example, is good research, or whether there should be other areas where the money should be going instead. Right, so moving on to the underlying biology. So this is the area where I kind of spend my days. Cancer is no longer a black box. So some of you guys out there, particularly the guys I imagine, will recognise this has been the wiring diagram from a 1974, I think, Triumph Spitfire. So, so those of you who understand wiring diagrams, it just tells you how a car is all joined together, and so where all the wires go to make things ping and bang and the wheels to eventually turn. So it's a critical part of a car's capacity to work, and it is also when it breaks, you will need to know your wiring diagram so you can go and find where the fault is and you can fix it. Okay, so it's critical. We're now getting a quite reasonable wiring diagram for the human cell, and therefore for cancer. It looks kind of like a little bit like this. This is a very, very simple version of it all, but these are called cell signaling pathways. So this is a cross-section through a cell, and the big oval in the middle is the nucleus of the cell, where all the DNA and all the genes reside. And so what happens in a cell is it has endless signals coming from cell surface receptors which listen to the world around that cell, and they send a message to the cell nucleus to say, we must divide, we must grow, we must move, we must, I don't know, maybe we should die, we should make some protein, some product. All that information streams in from hundreds and hundreds of receptors on the cell surface through these signaling pathways. And we've now got the point now where we kind of understand most of the pathways that exist, and a lot of them are shown here. But of course, they're way more complex than I've shown here. They're actually three-dimensional. There are numerous feeds into each of these pathways from many, many other sources inside the cell. But the basic structure is there. The basic wiring diagram is now available to us all. So we can now start to kind of think a little bit more rationally about how we design drugs to treat cancer. Because when a cancer develops, it's due to mutations in genes which are involved in a lot of these common pathways. So just to reiterate that point about these pathways, again, it's the idea of a cell which is receiving signals from the outside. It passes those signals down different proteins in the cell to the nucleus, where the genes are turned on, which govern how the cell will behave, be it cell division or migration, whatever the function might be. Mutations can occur in genes which encode all these proteins in these signaling pathways. And when these mutations occur, they can drive the pathway to be overactive and force the cell to divide, to proliferate, to migrate, and do all those things that a cancer does. It's just driven by the presence or absence of a mutation. So I'll give you an example or two of how this has been used to develop new drugs. So here's one of those pathways. It's called the RAS pathway. And so it's a cell surface receptor. So there's a little molecule on the surface of the cell which senses the world around it. And it will eventually get a message saying, look, it's time we divided. We have to divide now because our tissue is growing, we're getting bigger, we need to divide. And so a signal will come down through that pathway, it'll go to the nucleus of the cell, genes will be turned on which will make the cell divide. So that's all fine, a nice controlled process. But if you get a mutation in a gene called RAF, it will force that process to be turned on and turned on all the time at a very high rate. So regardless of the message from the outside of the cell, the pathway will be going bom, 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 firing all the time. And so the cell will divide, those daughter cells will divide, and you'll get a cancer will develop. Because there's a mutation in that critical pathway which signals normally from the outside of the cell into the inside of the cell. So the mutation is driving the cell to divide at a very, very high rate. But we know what the mutation is. So we know what fault has occurred and where in that wiring diagram in that particular cancer. So here's an example of a patient with advanced melanoma. And that patient had a mutation in the gene there, that RAF gene. So that RAF gene has mutated, and now that pathway is very active. And rather than the cell dividing occasionally every few days, it's now dividing really rapidly because that pathway has now been turned on at a rate which is way higher than is normal. And so the patient develops melanoma, and that melanoma spreads through the body. What some really clever scientists did was develop a drug called vemurafenib, which blocks that mutated protein and it stops it from working. It just sits there and it grabs it and just stops the action of that mutated protein. And fortunately, that was given to this patient here, and within about four months, you really could not see any obvious disease in that patient. So clearly something's going right? We've recognized a fundamental flaw in the signaling proteins in that patient's tumor. We've developed a drug which stopped that flaw, and the cancer has gone away, at least for a while. So we've used the wiring diagram, we've found where the wires are broken, we've repaired those wires, and the patient has got better. Obviously the story doesn't end there. This patient did progress, the disease did come back, and that is the problem that we have of drug resistance. Tumours can rapidly find their way around the drugs, and the cancers will usually come back. So now we're having to try and find ways to stop that occurring. So again, we go back to that kind of rational thinking. So if we have one drug which blocks the pathway and the cancer gets around that in time, if you use two drugs in the same pathway or two drugs in similar related pathways, then you can get a better blockade. So for example, that in most cases, treatment with that drug I showed you before, patients will survive about 6 months without any disease coming back. If you add a second drug in the same pathway, you can double the survival time for that patient. And so we're now starting to kind of get into this world of where cancer could be seen as being a chronic disease, where you can control its behavior, you can keep it pinned back for long periods of time by use of smart drugs based on the rational understanding of how the cancer develops, based on that knowledge of the wiring diagram of the cancer cell. I'll go with that one. So we are only part way through that process. So the estimates are, there are about 400 genes in the body which we know promote cancer. When they undergo certain mutations, become overactive, they will promote cancer. Of those, about 300, we think we can design drugs against them to stop them causing cancer. And so far, we've only designed drugs against about 10% of them. So we're only a small way down through our potential for actually stopping these cancers by our rational understanding of the various pathways involved in the cancer's initiation and progression. And this is really where there's a vast amount of effort at the moment, where we're starting to understand what the targets are, and we've got chemists around the world developing new drugs to kind of block these proteins which, when mutated, drive the abnormal division of cells and the growth of cancer. The fourth big area of development has been in the maturation of the field of immunotherapy, and there is now a class of drugs which, again, Chris will cover in some more detail, a drug called Keytruda. So they're called immune checkpoint inhibitors. So we have got this very powerful immune system which will recognize, often will recognize cancer cells as being abnormal. So the immune system has really evolved to recognize foreign invaders. So it'll recognize viruses, it'll recognize bacteria, it'll recognize fungi, and you'll get a nice inflammation response, and those bugs will often get stopped. But it also can recognize cancer cells as well. And so what it does, it will see a cell which has something abnormal on its surface, and it'll go, ha-ha, I don't like you, and it'll latch onto that cell. So the immune cells, called T cells, will latch on to the abnormal cancer cell. And normally, if that was a virus or bacteria, it would go, thunk, it would kill that cell. But what tends to happen in the human body is when a T cell latches onto a cancer cell, it binds to it and goes, you're abnormal, I've got you, then it stops. Because there's a signal on normal cells, which is the don't kill me signal. And it says, no, I'm not a virus, I'm not a bacteria, I am part of this human body, don't kill me. So it's a way to kind of make the immune system still useful, but to kind of make it not so active that it's always eating itself, so not always getting too many autoimmune responses. So we have this problem here where the immune system, the T cells, will recognize the cancer, which is great, but then the cancer said, no you don't, I am a normal cell, leave me alone. And so nothing happens. The cancer will survive, the immune cells will sit on the edge of the tumour, and they will not proceed on and kill that cancer. Until some very, very clever guys came up with some drugs called PD-1L inhibitors. Don't worry about the name, but that is the signal. The don't kill me signal is the PD-L1 signal. And it's simply a cell saying, no, I'm part of this body, I'm normal, I'm a human cell, leave me alone. What they've done is they've developed a drug which acts as a blockade for that signal. So the don't kill me signal is now pushed away, and is no longer reaches out to that T cell. And so the T cell now thinks, this is not a normal cell, this is a foreign cell, and it will go ahead and destroy that cell. So it's incredibly powerful technology. It's controlled clinically, I think. Chris might have a chance to clarify this, but probably you have vast numbers of steroids to kind of keep the immune system under control so you don't get a massive autoimmune response. So it's a bit hard to use clinically, I believe, but the power is extraordinary. You've now got an immune system which can recognize the tumour just fine, often, not always, but often, and now by blocking that don't kill me signal, the T cell can carry on and attack the cancer. So this is a really exciting technology. It's made vast improvements to the outcomes of patients with melanoma, and there's an incredible amount of effort internationally to try and find ways to extend its application to all sorts of other cancer types as well. Right, so that's really all I have to say. When the research carries on, that's because we care for those amongst us who are struggling through no fault of their own, because there we all go, but for the grace of God or the throw of the dice, because it is our shared cause and because we are winning. So I'm happy to take questions or pass on to Chris. Thank you very much.